Welcome to BioIT World's Trends from the Trenches podcast, your insider's look at the science, technology, and executive trends driving the life sciences. I'm Allison Proffitt, editor of BioIT World. This episode is hosted by Stan Gloss, founder of BioTeam, a life sciences IT consulting company at the intersection of science, data, and technology. Joining Stan today is Andrew Hopkins, founder and CEO of Exscientia. The two talk about how changes in the drug discovery process will fuel a revolution in the industry and Exscientia's vision of AI-enabled drug development. Let's listen in.
Well, good morning, Andrew. I'm so excited to talk to you today. I've had this one circled on my calendar for quite a while, and we're finally together.
Great to be here, Stan. Really enjoying this. Looking forward to the conversation.
Yeah, it's always special for me as a former CEO of BioTeam and co-founder to have a conversation with yet another founder and CEO of another company, and being able to kind of resonate between the two of us of our kind of war experiences and stuff. But a lot of people might not know who you are in your company. So I'd love you to kind of introduce yourself to our audience and, tell us about yourself and the company and all of that. And then we'll start our chat.
Excellent, Stan. So as you know, I'm Andrew Hopkins. I am the founder and CEO of Exscientia. Exscientia means from knowledge in Latin. And we picked that name because I thought it was really describes the philosophy of what we're trying to do. How do we create better drugs from knowledge and all of the information and data that we have available to us now? My background is as a scientist. Actually, I've always worked as a scientist, even from the age of 16 when I was doing my internships in steelworks near where I lived, actually, and got a scholarship from them eventually for my work there. And that helped me through university as a chemist at first. But actually, I also had a really strong interest in sort of computational approaches, even sort of doing computational chemistry in my final year, etc. And that's when I really, really sort of started to see the possibility of computers and drug design starting to come together, actually. This is probably like, early 90s now at the moment.
Wow.
And so, and I knew I wanted to move into that space. So, but actually, since I had a scholarship, I went back to the steel industry for a while, actually, for a year. And that was quite an interesting experience, actually, very formative, actually, in the sense of really helped me understand the creative, destructive power of commerce and industry. Mm-hmm. And what I mean by that is I used to work at a steel factory in a place called Port Talbot in South Wales. Absolutely huge. If you go past it by train, it takes about 10 minutes to go past. It's an enormous place. And in a lot of late 80s, we had nearly 30,000 people working at that plant. Huge number of people, from the whole town or area. They weren't working in a coal mine in the 70s or working in the local sort of tin plate factories of an oil refinery, very, very industrial area. And then what happened is that new technology came in, new ways of working, new processes. And what we led to then was a reduction of about nearly 90% of the workforce, from like 30,000 to 3,000.
Wow.
And a 20-fold output in productivity per person, sort of tonnage per person, etc. Of course, this had devastating social effects, a lot of unemployment then in the 80s, but then you also start to see, this is an industry which has really tight margins, really thinking about, well, how it needs to innovate to stay ahead of the game, etc. Particularly at South Korea, China's starting to come online, things like that. As it's a tough industry. But actually, those lessons, stayed with me. So eventually, when I ended up in pharma and biotech, I think I had a very different perception of the power, of commerce and markets and what they can potentially drive, and the power of new technology as well to position oneself in those markets. And I never actually saw things as standing still. And whereas other people might have thought getting a job in a big company was like in tenure, which almost was like in the big pharma in the late 90s, early 2000s, it was almost like getting tenure in the university professorship. But I had a very, very different perception of it, actually. And that stayed with me all the way I've started to think about really the intersection between economics and the industry we're in, drug discovery and development.
Mm-hmm.
Anyway, so following that, then I was fortunate enough then to do my doctorate at Oxford in HIV drug design with Professor Sir David Stewart, and who was a real lead in light in crystallography in the UK and virus crystallography. And that was an amazing period. I would, if you caught me when I was doing my PhD, I would just absolutely bore you senseless completely about how excited I was about working on HIV in a field.
Mm-hmm.
And if you go back to that period, it was a period, don't forget, when we started in the sort of the mid-90s, the only treatment we had was AZT, which wasn't really a very good drug. And then we had a whole new generation of drugs, non-nucleosides started to come along, and protease inhibitors, and the whole sort of mathematics of understanding the virus evolution and combination therapy. And it allowed us then to really put into context how we would then start to, think about cocktails of drugs and how we would design drugs against mutants. And in fact, that work really, I think, influenced me for life then. It really got me at the interface then between drug design, structural biology, crystallography, biophysics, and computational approaches, which we were developing, working with Peter Goodford, who was one of the early pioneers of computational chemistry, who had a program called GRID. And working with him then directly and thinking about then computational approaches about this field, even getting into the lab and making my own sort of compounds I designed and getting them tested and seeing those results, running your own assay, compounds you synthesized, doing that whole sort of little mini biotech in your own PhD sort of thing. And that really just gave me a huge interest then in drug discovery, how to do that. Interesting side story, the guy who taught me, chemistry at GSK when it was Glaxo Wellcome at the time, this guy called John Milton, who later on went on to become the CTO of Illumina at Oxford Nanopore. So it was quite interesting just to meet these people along your journey, And and for ages I actually didn't see him, and he said one of the best quotes I've ever heard of someone say. He said, "John, I hadn't seen you publish for 10 years. What were you doing?" I was too busy inventing the damn stuff.
Yeah. So as you described that whole path towards the HIV research, I was thinking of a book that I recently read called A Shot to Save the World, and I'm trying to think of the author's name, but I can't this second. But it actually traced back the messenger RNA all the way back to the early research into AIDS. And so some of the research that was being done on AIDS back in the 80s are now come full force and helped us get to a vaccine in the production of it. The technology of messenger RNA has been around for a while, getting it produced. So that was really cool. I also was thinking...
I used to work on reverse transcriptase myself, which, as you know, it converts RNA back to D, and it's DNA to RNA, and it's and of course that was a key drug target, and it was the mutations of that which led us then actually to really think about, an insight that led to Exscientia fundamentally. I've told this story before, but I'll say it again. Actually, it was that sort of interface of thinking about drug design and what it takes to create a drug, and sort of the firmament you're in that sort of field of HIV, which probably was similar to the field we were in recently with COVID. That sort of energy to create a drug, quickly, and the urgency one felt. That led me walking home one evening about 2 a.m. in the morning. To think about there must be better computational ways to think about how is a drug created. And that idea, there must be a much better computational approach to thinking about how to design drugs, stayed with me ever since. I remember that moment for, up until this time. And all the way I went through after my PhD, or DPhil as they call it in Oxford, went to Pfizer, which was an amazing place to learn the art of drug discovery and development. And but it always struck me that there must be a better way, because then that led me then to really thinking about always having sort of interests in data and informatics approaches as ways to solve these problems. There's a great quote I came across actually, which is the pharmaceutical industry is an information industry. And when you break it down like that and think about everything we're doing is about data generation, data analysis, and analyzing that data to make decisions. And actually, it's true. In fact, if you look at that little white pill people take, we can argue how much it may cost to manufacture, which is a fraction of the cost often we sell drugs are sold for. But it's not the manufacturing costs; it's the IP costs, it's the cost of invention, the cost of attrition, etc. That's what's driving that, and that molecule that we've manufactured, that drug you now take, it's been tested. The huge amount of sort of intellectual work that's gone into creating that data package, but also the possibility it's that one molecule out of almost an infinite other possible choices it could have been. I thought was an extremely interesting question. How do we then ultimately convert what is then an information industry then into using modern compute, modern informatics, modern machine learning approaches to then help us make much better decisions, to make effectively run this as a much more effective industry than it is actually. And I think actually we have a moral duty to try to run the most efficient, effective industry we have actually, given health. Good health is ubiquitous. Everybody wants it. We want to try to cure diseases which can't be cured. And we want to do that at a scale to try to help as many people as possible. So I've always felt there's been quite a moral need to think about how then we improve the ways we can actually, invent new medicines.
Back when I started in the industry, pharmaceutical companies were based on chemistry. Chemistry based, it was all compound libraries, and all doing all kinds of work around chemistry. And that now is pivoted over. The supercomputer and Amazon cloud and other types of technologies are all part of the laboratory of a scientist now. So much of this has become computational. There's a quote I wanted to share with you that I recently interviewed people at Recursion Pharmaceutical, and one of their chief product officers.
I know Chris very well, actually. Yes.
Yeah, I just actually his podcast was just released on Tuesday, so you could check out my interview with Chris. But Mason Victors, who works for him, I asked him, what's the difference between this nimble startup companies that are doing digital drug discovery versus traditional drug discovery? And he said that traditional drug discovery views the data as the exhaust of the drug discovery process versus us that look at it as the fuel to drug discovery. So what's your thought about that approach? And do you see that in the market?
Absolutely. That really chimes the bell. For when I think about when I started building our first large-scale information systems and data warehouses at Pfizer, I was interested in the concept of how could we learn from basically all this almost waste material, as you call it. People would run a project. We'd use the data at that moment in time for making a decision, which compound to make next, for example. And it basically then sat in silos, almost gathering electronic dust, so to say. And it was then I realized if we could integrate this data together, and it's that then led me into understanding the field of ontologies and data integration and things together. But it allowed us then to realize we could create new knowledge from the integration of all these fragments of data. And that's an important thing. It's that sort of T.S. Eliot hierarchy of it between, where is the wisdom we've lost in knowledge, where's the knowledge we've lost in information, etc. And so it allows us then and we published a paper in Nature Biotech back in 2006. I think this was one of the very first papers which really showed large-scale machine learning on pharmacological data. And we were only able to do it because we did this huge data integration exercise before that, which allowed us, don't forget, Pfizer in those days was a real accumulation of several companies, busy doing mergers. We got all these new companies coming together, all these new sort of data sets. How could we then, take our raw data, integrate it all together at the chemical level, at the screen level, at the target level, etc., and understand that data? And what it gave us then was, we call this paper the global mapping of pharmacological space. And then so rather than see these individual silos of data or these historic projects, when linked together, we could see then the interrelationships. We could see polypharmacology for the first time. Hey, did you know this compound that you made in 1984, it was also active against this other project in 1998, and it's actually a pretty interesting lead for it, etc. And it allowed us to understand sort of the network pharmacology of those relationships. The other thing it gave us, it actually allowed us to aggregate our data together to give us, really good size models. Good size data sets then for building large scale machine models. I was doing that with a colleague, Willem van Horn, who also works at Exscientia. He was one of my early sort of conspirators in building Exscientia. And it allowed us then to really build like hundreds of these machine learning models in parallel. And then we could start to do virtual screening and virtual sort of polypharmacology predictions, etc. And I think that was one of the very first papers that did that on sort of a large scale. And that actually was one of the foundations. It was fascinating because we're working with actually my old boss, John Overington, who actually hired me into Pfizer. These are the days when you could just hire someone because you like to cut a jib, so to say. I think I was hired almost without a job description. Just come work here, And John went off to work for a company called Informatica, and he was doing something very interesting of like, he didn't have the data, which we had at Pfizer, so he need to create his own data set. So he was actually commissioning, curators, taking all of JMedChem, all of the pharmacology and medchem journals, converting this into machine readable format. So we integrated the external data as well as the internal data. And back in 2007, and about a year later, I left Pfizer to become a full professor up in Dundee. And the very first week on the job, where I also knew I needed the data, I remember writing to the Wellcome Trust and going to see them down in London with Janet Fortin and saying, you should actually buy this data. We should make it public. And that became the Kemble database. And John then moved from Informatica, which is then part of Galapagos. And a year later, Kemble was formed, which became, I think, one of the foundational data sets then of linking chemistry and pharmacology and biology and genes to allow us then to start to build this new industry, because a lot of this stuff, great data in pharma, and only now they started to realize how to, create knowledge from that data. But importantly as well, I think what's been a real spur is these large open source databases, which then came online, from around sort of 2008, etc., which then allowed us, myself included and other people as well, like Chris at Recursion, to then start to have these large open sources data, which we can then build machine learning models on, try out new technologies. And it did become a big spur. I think it's a good story of how, public sector investments actually then can spur innovation in the private sector by giving us sort of an equal foundation, which we can all build upon.
So let me ask you, your company, why did you found this company, and what was the burning reason you saw? Every entrepreneur says, has a reason why they start their company, either to do something better or a crying need or something. So what was that, and why did you do it?
That's a great question, Stan. And it's something that's been on my mind for over 20 years, actually. It wasn't just a whim. It was something I've been thinking about for a long time, in the real need and desire to change the mechanics and the process of how we invent and create drugs. It's not enough actually to just, invent a drug. I think we also need to think about the process of how we invent drugs, how we invent new ways of doing it. It's been said before, but I'll say it again. It's think of the car industry, in the US, over 110 years ago, hundreds of car companies, hand-building cars, just like Morgan cars still do today in a way. But the real innovation was not inventing a new car. It was actually inventing a process of how to manufacture cars en masse. And that's what Ford did. That's what led ultimately to bringing the price down, so a car in every yard.
Right.
And that in many ways is how I think about our industry. We've built a great model. Pharma's a certain way of producing drugs. Biotech had a certain way of producing drugs. Raise money from a VC, go take it to the CRO. CRO gives you a drug, gets more VC money, take it into one of the CRO companies to run your trial. Great way of owning an IP, great way of developing it, etc., but not necessarily changing the fundamental process of how we change this industry. That idea has been in my mind for a long time. So it's driven by that. And the other thing as well, once we started to then think about starting to see the emergence power then of machine learning on large scale data then allowed me to think about, well, what if next? What if we could then invent molecules this way? And that's why I really, when I went to Dundee, that's what I went to do actually in a way. It was, I saw being a professor as a sabbatical, so to say, to have the time and freedom to then think about this as a problem. So a problem we were initially interested in was, how could we create a sort of a model-driven approach using machine learning to actually, design drugs? You know, one of the problems we have with drug design is you have a lot of data streams coming in, a lot of different data, and then you chemical imagination thinking what compounds to make next, etc. This is a process that takes many cycles, lots of iteration, sometimes thousands of compounds being synthesized over several years as we learn about the target, learn about the chemical space, etc. But as I said, if you think about the industry as an information industry, you think about it as a problem of data and decision making, then it allows you then to start to think about how then we could have better ways of integrating all the data together. That's what I saw that machine learning models could give us. And then actually, how could we then think about mimicking the creative element of design? And it's fascinating now seeing all of the work with, generative AI coming on board in terms of fields of art and writing now with ChatGPT. But it's the concept of generative AI, which actually has been on my mind for 15 years. And that's what our original work actually, which we published in Nature back in 2012, was about. How could you use generative AI approaches to actually mimic the creative aspect of drug design?
Right.
And in those days, we were very much driven by evolutionary computing approaches to doing that, combined then with machine learning models. And the analogy that led me to that sort of architecture was, one interesting analogy for the creativity of humans is it analogous to evolution? The idea of generating many ideas and then actually selecting those by some sort of function, fitness function. If you're an artist generating ideas, you might come up with many ideas, but your fitness function is your concept of beauty, what actually would be that. So in ours, actually, our fitness function was the models we could build for different biological activities, the targets we want to hit, the targets we want to avoid, and the other sort of PK properties we want. So those became then our fitness functions, which we could then explore using generative AI then to find it. And we showed then in that paper that you could actually target quite exquisite, polypharmacology or selective profiles across, over 20 sort of receptors to say, fine tune this kind of activity and generate novel IP, which was then patentable in its own right, via algorithm. And that was the big breakthrough, showing that stuff, molecules then obviously in vivo active, etc. And so that then led to the foundation of Exscientia. So we started off by this first problem of how can sort of AI, generative AI approaches and machine learning help us potentially to design drugs using this kind of approach. And that's how and when we started Exscientia, originally we started it, working with the university to create a spin-out company for my lab. Jeremy Besnard, who I was working with, was my PhD student, was our first employee, along with Richard Bickerton, my postdoc, both of which still work for the company today. And it allowed us then up in Dundee in Scotland to then, create this new company. And originally started it, on my credit card, effectively buying our first AWS cloud computing and laptops.
That's amazing.
And in fact, the advent of cloud computing at that time was a key instrument in allowing us to sort of start the company up quite cheap, because, some of my friends had previously formed companies and needed to raise their first $1 million just to build the big iron, the big compute farms.
Right.
But now this was available, on a credit card, on the cloud, buy as needed. And that was a huge innovation, actually, in changing the economics of a startup, particularly, obviously, a computational-driven startup. So that was an important sort of feature as well. The other feature, since I'd been at Pfizer, since I'd been publishing a lot, I was, those days fairly well known as a scientist in the field. But it allowed me then to make contacts. And we actually started Exscientia in a very unusual way. We actually, avoided VCs. I used to advise, VCs, a huge respect for them. But I wanted to build a company for the long term. What do I mean by that? I wanted us first to prove a technology out before sort of scaling the company. And so I started off with deal making and doing deals with early customers, like Salvian, almost from day one. And that, because what was important to us then was really testing the technology out on real world problems. And that, I think, is you need to innovate at the cold face, really. You need to see how the technology is going to play out in the real world and understand. And that was important, because what we were gaining then from those early collaborations, it was the data on how our systems were performing. And that was actually vital to us then, actually. That's how we could then be exposed to different problems, then think how we could quickly iterate and develop new technologies and for new problems we're starting to see then.
So maybe you could share a couple of stories of maybe some of the clients that you've served and how you've helped them. I know that you've had some press releases around some of your strategic clients that you serve. Maybe share with us what's public, obviously.
Sure, yes. I mean, when starting a new company, particularly with a new technology, you really need sort of vanguard customers as much as inventing a new technology needs an entrepreneur to drive it. The two sort of live in a symbiotic relationship. What I mean by that is, if you want to test out new technology, we were at the sort of cutting edge and actually proposing something quite challenging. That here's something as creative as drug discovery that actually an algorithmic approach can make a challenge to, could improve, help upon. But actually, a lot of people did see it as a challenge as well, almost an existential challenge. And I think we started to overcome that when we talked about the centaur concept. It really is about sort of combining drug discovery experts with the advantage of a machine. And this came from the concept of centaur chess, advanced chess, that people like Kasparov had been demonstrating. And there's some really interesting sort of analogies we found in that field, actually, which was the concept of, someone using the right algorithm in the right way, machine plus human expertise plus the right process, that a master could start to, challenge a grandmaster, and it equalizes skills to a degree. The other really interesting thing we found out about many of those games was people stopped making mistakes. The whole level of skill set generally started to improve as well. So what it was about then, but importantly, a lower machine could beat a human. We also started to see that a human plus a machine could beat a machine and beat a human. So there is still then this concept of combining the different skill sets, the tacit knowledge, many of the things that machines can't do yet, alongside the compute power of a machine as well. So this concept then of we are better together, the human expert plus the use of advanced generative processes and machine learning models can actually hopefully create a better process. And that was actually a key factor. And so we had a few key advanced customers that recognized that. But importantly, pharma is a data-driven industry. So we wanted to make sure
it is now, for sure.
To these are not claims, this is not hype, but we actually got the data to show. Exscientia has got that data. Hopefully very shortly we'll have our fourth drug, created by our platforms, and our teams entering into the clinic. That's fantastic. Now, the first company to have truly AI generated molecules now in clinical trials. First company to show we also now can show that an AI system selecting drugs based on sort of single cell analysis of patient material can improve clinical outcomes in oncology, and the clinical trials now EXALT-1, which we've now published to demonstrate that. So we start to show now that AI can start to make a difference. And some key stories, I think, actually, is different problems we might have solved for people. One of our early partners was Dainippon Sumitomo Pharma in Japan. And we were working with them on the concept of bispecific small molecules. As you know, often we're developing a drug to target a particular protein. Here, very interested, could you actually take our polypharmacology idea and design a drug that deliberately targets two proteins at the same time? Could you design a drug that's an agonist against one and an antagonist against another target? And we managed to do that, actually. We've run 3 successful projects. 2 of them entered the clinic already. And so it allowed us then to really demonstrate the system could generate those and generate them actually in a very efficient manner. We were only making around 350, 400 molecules to create these bispecific small molecules. And we did that in around 12 months. Wow. And those are stats which we've continued to improve upon since. Some of our latest molecules, we've actually, CDK7, for example, it's heading to the clinic now. That was less than 150 molecules, new compounds synthesized and tested. In starting a project to identify the drug candidate, about 12 months, or less. And we've got a lot of these robust, done this now for about 8 drug candidates, where we're getting very similar kind of statistics. We're effectively making, I would say, around 90% fewer compounds compared to industry average. Industry average often is about 2,500 compounds sometimes in a drug optimization project. And so it allows us, and we've done that actually with this combination of how do you combine experts with AI in better decision-making and design. Right. I love this quote, again, from a systems thinker called Robert Akoff, that knowledge is the efficiency of choice. And what I like to think is that as we create sort of new concepts of knowledge from our models, that actually the efficiency of choice is actually as we are then, becoming more efficient in how we optimize is because hopefully we're making better choices from better knowledge. And that's how I ultimately see this connection between knowledge and efficiency in what we're trying to do in our optimization projects. Other great ways that we started to collaborate with people. One of our, really important deals in the history of a company was when we signed our first deal with Celgene, which later became then BMS. This was a 3-project deal. And, we've already had a first project of that licensed, the lead molecule. That was an immunological indication. Hopefully more news on that particular project moving forward into the clinic actually this year. Yeah. And actually great news as well, hopefully be announcing shortly on the other molecules we've been developing. And so we started seeing that was really important for the company because it allowed us then to move from being this sort of just sort of a design company, and purely an in silico company, to one then where we then started to realize that generating our own data, running our own projects then became sort of a new key skill sets we developing. So we integrated my wife's company, Kinetic Discovery, into Exscientia. That then gave us sort of a structural biology, biophysics basis then for building out our pharmacology. We then started to bring in some great, more drug hunters to help us then run the projects. And the skill sets required then for managing the complex relationships between your own teams, CROs, etc. And then we moved then from just producing intellectual IP to a company then that's really producing the full data sets, running the full projects, producing the molecules then that become the INDs. That then set Exscientia then on the new trajectory to become the company it is today. And that was a really important deal actually with BMS. And BMS continued to be a very deep and important partner for us. Just last year, we expanded our deal with BMS, going from 3 projects now to 8 projects. And that allows us then to continue to build our relationships and hopefully bring more projects forward with BMS in the future. The other really important one was a collaboration we've been doing then with Sanofi.
Tell us about that.
It's started off actually as a small project, a single project collaboration. And this was actually an idea they came to us with, again, around a sort of a bispecific small molecule. They wanted us to know if we could design a specific enzyme inhibitor that was hitting 2 very, very different enzymes at the same time, completely different gene families, And we actually managed to do it. It was a huge calculation. We were exploring quite a large space of possible targets in the area of sort of immunology and fibrosis. And I remember over one weekend, we ran a calculation where the generative AI was generating something like 100 billion molecules against a whole matrix of potential targets. And from that then, we made literally a handful of compounds. And we found these lead molecules, which had this nanomolar activity against both of the targets we wanted. So we truly had this bispecific. And that actually led to an early licensing deal where that particular project then was licensed into Sanofi.
No.
Obviously, that, I think, actually won us some brownie points with Sanofi. So a little bit later then, we then started to build up our relationship with Paul Hudson and Frank Nestle at Sanofi. And we could then see the possibility of what we could do at scale. By now, we'd already started seeing we were delivering for BMS, etc. And because I think we'd already should have shown our mettle in this solving this hard problem earlier, that led us then to think about actually a much more ambitious collaboration. How could we bring the advantages of what we were building with AI approaches and combining that then with expert biological data generation to allow us then to think about how could we build a pipeline actually together. And that then led to a deal we signed last year, which is $100 million up front and $5.2 billion in downstream milestones and some very, very significant sort of royalties as well associated with those. But it allowed us then to really think about how we could then, sit alongside and partner with Sanofi then to really think about how you use the scalability that we think is potential in this technology to actually not just try to then create, projects and great molecules, but also even how do you then use it to scale to build pipelines and we'll work together to do that. And that's what's really exciting about the ambition of that deal.
Well, these are really fantastic stories of, just the whole journey that you've taken from starting a company with a credit card and building it up to working with Sanofi and BMS. I have one question for you. For the entrepreneurial scientists that are out there in labs and doing their doctoral dissertations and all of that, what advice could you give them about, kind of combining their scientific background, the scientific method with the entrepreneurial method, which actually are quite similar? And what advice could you give to a young entrepreneur today who wants to listen to this and follow in your tracks?
Oh, God, that's I feel very responsible, actually, the advice I may give them. The way to think about it is make sure you have a deep vision that you believe in, because this is a long journey. And the ones who often survive, the ones who make it, are the ones who actually stay the course. And it's almost like the survivors' luck in a way. So you need to think about, is the idea big enough? Is the idea, have you got actually the internal character that you are prepared to stay the course on this journey? Because there will be many, many winds that try to blow you off course along the way. I think the other thing is as well is always be fleet of foot, having an open mind, thinking about the opportunity that is ahead of you. So you need to think like both a fox and a hedgehog. You might know that sort of that parable. And so the idea then is, I think you need the hedgehog concept of the one big idea, stick with it, to go with it, etc. But you also need the mind of a fox to make sure you understand when a pivot may be required. You understand the opportunity that's emerging. And the other thing is to absolutely learn from others. It's not about the entrepreneur. It's about the team. It's about the people you bring with you, the people you could all realize you're all in this together. It's you against the world about how you want to build this. And your team is absolutely vital, and make sure you bring them along and very well rewarded for it. And because that's what's going to do it. This is never about an individual. This is always about how a team actually creates these things. And that's key to it. And your team is as very much your customers. Understanding what you can learn from your customers. We've learned more from our customers than we knew ourselves and taught ourselves. And be open to because that's what we really want to do. It's your technology will only get better when it's interacting with the real world. The flaws you see are the opportunities to make it better and to develop it. And to have that responsiveness is absolutely key. Because in many ways, you may well be heading towards that vision you want to get to, but it isn't a straight road. It's actually how you understand and navigate the paths along the mountain as you go up there, which actually I think makes it a successful climb.
Yeah, for me, it's, understanding that you have to love your clients and love your staff equally along the way. Well, listen, this was a fantastic conversation. We could do this again another time, but this was terrific. I loved your journey and really appreciate you sharing it with us today.
Oh, thank you much, Stan. I feel that's only chapter 1 of our journey. Chapter 2 we write in now is how AI is actually going to change the type of molecules that we invent in a kind of drugs. And I look forward to telling you that one day about how actually AI is going to really bring precision medicine to the people.
We will absolutely do that. Thank you so much, Andrew.
Thank you, Stan. Great to chat to you.
Cheers.
Thank you for listening to BioIT World's Trends from the Trenches podcast.